Welcome everybody. Thank you for joining. And I want to welcome you all to this session of the session 6 on microbiota and targeted nutrition. For a long time, we know that the microbiome is important, but what I'm going to share with you and introduce our speakers is the idea that in health and disease, there are very important things to know about the microbiome. We set up a big birth cohort study in Canada, the largest in North America, the child study, to study gene and environment interactions in health and development. And of course, a key factor is microbiome in that health and development. Many researchers have gathered together with their students to share data from meconium through the early years of life. The composition of fecal microbiota in healthy infants is really both the phylum and the types influenced by the mode of delivery and whether within each mode there is breastfeeding or not breastfeeding. So even right from the beginning, in healthy infants who are born, the mode of delivery and how they are treated over the first window of time, and in fact in the first 3 months, is extremely critical. This slide shows differences in the microbiome between controls and those who end up developing atopy and wheeze in the later years. Already at 3 months, there are differences in the microbiome, especially in these 4 species of bacteria. And that is an extremely important observation because it says critical windows are open early on. In an animal model, published also by child scientists, they could abrogate the inflammation, the cell counts, the types of cells, and the microbiome by adding back those deficient species that were found in the humans and abrogate this course of events that leads to airway inflammation. And the same is true for the kind of feeding, formula or not, the types of things we add to, whether they're pro or symbiotics, and how the microbiome develops in the first 100 days, its richness, its diversity, would be extremely important in determining the trajectory later on. So what we're going to hear about today is about altered and changing and nutritional ways to change the gut microbiota towards health. And this relates not just to allergies, but to adiposity, obesity, and cardiovascular disease. The mother's milk, the infant microbiome, the type of feeding, the type of bacterial and other environmental co-occurrences will all play into this. And I hope we will learn after this session something about how to approach the microbiome from a health and wellness point of view and a rectification of disease point of view, prevention even. So I'm very pleased to introduce our speakers today, Dr. Karina Venter, who's going to speak about manipulation of the gut microbiota in early life with symbiotics, which infants benefit, and Dr. Professor Anna Novak-Vegin, who's going to speak about the clinical benefits of symbiotics beyond dietary management. So with that, let's start. Hopefully we'll have some time for questions at the end, but we'll go through the talks. And now I will introduce Dr. Venter. Thank you all for joining us today.
Good morning. My name is Karina Venter. I'm an allergy specialist dietitian from Denver, Colorado. And today we will be discussing manipulation of the gut microbiota in early life with symbiotics, the infant that benefit. please take a note of my disclosures. We will also discuss today about who is at risk. So who is the infant at risk? What is a normal balanced microbiota for a healthy breastfed infant? What health outcomes is this associated with? What is an imbalanced GI microbiota in infancy? What factors are associated with an imbalanced GI microbiota in infancy? What are the clinical implications of an imbalanced GI microbiota in infancy? Is it possible to modulate the GI microbiota? And then I will summarize with a few key points. So in terms of family history of allergy, maternal history of allergy, parental history, and paternal history of allergy, there's been quite a number of publications. But if we could just focus on food allergy outcomes for now, then we can see that maternal history of allergic disease, parental history of eczema and asthma, parental, paternal, or maternal history of atopy, and also maternal history of food allergy put an infant at risk for developing food allergy. So what does a healthy infant gut microbiome look like? We have to take into account that it's not a matter of one size fitting all, and that the infant gut microbiome will be affected by mode of delivery, whether the infant is breast or formula fed, the maternal diet during pregnancy, and perhaps maternal diet during lactation, and then also other environmental factors, and I will get back to these a few slides further down. But if we could for now just focus on what we see in terms of the infant microbiome in breastfed versus formula-fed infants, we will see that the breastfed infant microbiome is more stable. It's more bifidogenic, as you can see from the levels of actinobacteria, as bifidobacteria is an actinobacteria, and it has less pathogens. Whereas with the formula-fed infants, we see that the microbiome is less stable. This will also very much depend on the infant formula that the infant is being fed. There may be more pathogens. And then to summarize from this slide, we can say that breastfed infants had more total proteobacteria and bacteroides, and the formula-fed infants had more firmicutes and no bacteroides. So in terms of health outcomes of a healthy microbiome, so why do we want a healthy microbiome? What is the health outcomes going to be? And when we look at a healthy microbiome, we say that it is characterized and balanced with lots of bacterial communities, with imbalanced gut microbiome, or often referred to as dysbiosis. We see that the gut microbiome is poor and unbalanced in terms of bacterial communities. And then really to summarize, I thought I would show you this diagram where we assume or we postulate that a healthy diverse microbiome based on the available evidence on the moment produces more short-chain fatty acids, like for example, butyrate and propionate. With more levels of butyrate in particular, we see more T regulatory cells, and that is also shown by studying the cytokines, and infants with more T regulatory cells have more T regulatory cytokines such as IL-10 and TGF-beta. So in summary, really, we could say that an infant with a healthy or diverse microbiome is more prone or is less prone to develop allergy, and we see less inflammation too. So really, in summary, healthy microbiome, more diverse microbiome leads to more T regulatory cells and their cytokines, and therefore less allergy outcomes. So what is an imbalanced gut microbiome in infancy? This microbiome is less diverse, has more pathogens, and there's an abundance of unwanted bacteria. And so here on the slide, you can see that a good microbiome is characterized by more bifidobacteria, E. coli, and lactobacillus. So we definitely see differences in infants with a balanced or an imbalanced microbiota. And the imbalanced microbiota has got more Campylobacter, Enterococcus, and C. diff. So factors affecting the gut microbiome. This is a redrawn figure from a paper by Nonhlanhla Lunyani, beautifully summarizing which factors can affect the gut microbiome. And factors that we've, of the infant, and factors that we've looked at in the past has been genetics, prenatal exposure, particularly maternal diet, delivery method, feeding method of the infant, also the infant's diet, drug use, particularly antibiotics, and we will get to that again a few slides further down, housing conditions, environmental exposure to pollens, family structure, smoking, and then also animal exposure and season of birth. So really a large number of factors that eventually will define the infant gut microbiome and then disease outcomes further down. But one of the main factors affecting the infant microbiome is method of delivery. And if we compare infants born by C-section versus those that was normally delivered, we can see that the vaginally delivered or normal delivered infant has more bifidobacteria, similar to what we see in the breastfed infants, and less Clostridium difficile. Whereas the cesarean delivered infant has got less bifidobacteria, so opposite from what we see from the normal delivery, and they've got more Clostridium difficile. So definitely differences seen, and the gut microbiome in the cesarean delivered infant is also less diverse. Interestingly, pollution may affect allergy outcomes. So pollution, we know, has been associated with development of wheeze, and I think that has been well established and described in many papers in the past. But recently, we've come to learn that the indoor and outdoor pollution can also affect the gut microbiome of the infant that's being exposed. And it affects levels of the bacteroidetes, firmicutes, and also the microbia. So definitely we can see that the bacteria can affect the gut microbiome. And in particular, I've already highlighted the importance of the short chain fatty acids in a diverse gut microbiome, where butyrate is one of the most important short chain fatty acids to lead to T regulatory cell functions. And with indoor and outdoor pollution exposure, we also found less bacteria in the infant gut microbiome. So definitely factors to take into account. So what other factors are associated with an imbalanced gut microbiome? I previously mentioned that we will get back to drug use in infancy, and here you can see that there's differences between infants fed or given antibiotics, proton pump inhibitors and antacids when we look at children with cow's milk allergy, peanut allergy, and egg allergy. So clearly, early use of antibiotics, antacids, and proton pump inhibitors in infancy can affect allergy outcomes too, and via its effect on the gut microbiome. What are the clinical implications then of an imbalanced gut microbiota? So many studies have been published looking at gut microbiome and infant disease or childhood disease outcomes. I'm going to focus just on allergy outcomes, and here you can see that allergy development has been associated with lower lactobacilli and once again lower bifidobacteria, I've highlighted that a few times now, and lower bacteroides, increased Clostridium difficile, and a lower diverse gut microbiome. So we have a lot of work to do to see how we can best change the infant gut microbiome to prevent allergy outcomes. So the infant microbiome and sensitization. This is data from the child study with the first author, Megan Azar, clearly showing, if we just look at the first figure for now, because it is a busy slide, that in infants that sensitized to food allergens compared to infants not sensitized, we can see clear differences in many of the different bacteria study. And in this figure 1, you can see that the non-sensitized infants have more bacteroideia, and those sensitized have lower levels. And that was similar at 3 months as it was at 1 year. So once again, showing that even before we get to clinical disease, just looking at IgE production and sensitization, there's clear differences in children who become sensitized at 3 months and again at 1 year versus those that did not become sensitized. Then what about children who outgrow food allergies? You know, is there a difference in the gut microbiome of children who remain food allergic versus those who will eventually outgrow their food allergy? And this is data from the Mount Sinai group, from Supinda Bunjavanic, showing once again clear differences between children who outgrow their food allergy versus those who remain allergic, with those that eventually outgrew their food allergy showing a higher diversity of the gut microbiome at 3 to 6 months, 7 to 12 months, and then again 13 to 16 months. At this point, it wasn't significant anymore, but you can still see the slight differences. So how do we manipulate the gut microbiome? Then, you know, for disease prevention and also allergy and food allergy prevention in infancy. We can use prebiotics, which is defined as a substrate that is selectively utilized by host microorganisms, conferring a health benefit. That's really the good fibers in fruit and vegetables and in some grains. We can use probiotics, which is the live microorganism, so the good bacteria, and when administered in adequate amounts, confer a health benefit on the host. Or we can use the two together, which is referred to as symbiotics. And the symbiotics is a combination of the microorganism, so the probiotics, to have some beneficial effect when consumed, and a compound that specifically favors its growth, a prebiotic, so the good fibers, having a synergistic effect when paired together. So it seems that probiotics have got some beneficial effect, and prebiotics have got some beneficial effect, but we wanted to study to see whether if we give the two together as a symbiotics, whether the health benefit and the effect on the microbiome may even more pronounced or clearer. And so when we look at this study using a standard formula with symbiotics and the microbiome, you can see clear differences between the children in terms of bifidobacteria composition, so the in the gut microbiome in children that was given the normal formula versus children that was given the formula with the symbiotics. And the two groups of children also had a C-section, the control group. Here you can see the gray dots, and the C-section with the symbiotic group. There you can see the increase. So just comparing the two children with the two groups with C-section, you can see lower levels of bifidobacteria in the control formula and much higher levels in the children that's having the supplemented formula. And then if we compare them to the third group, which was the vaginally delivered or the normal delivered infants, we can see that the C-section delivered infants with the supplemented formula had a gut microbiome or bifidobacteria level much closer to those infants that was born by normal delivery. And then, so we can see the effect now on this slide on the microbiome, but what is the clinical effect? And we can see here again that when we look at the symbiotic group, we had less skin disorders, and we had a 72% reduction in atopic dermatitis or eczema. And so the conclusion was that given a symbiotic added to a standard formula can change level of particularly bifidobacteria in the infant microbiome, but it also has the potential to reduce the development of atopic dermatitis or eczema, particularly in the C-section delivered infants. So if we then look at the effect of using the symbiotic with an extensively hydrolyzed formula, once again, we can see higher levels of bifidobacteria, lower levels of Clostridium bacteria, and in terms of the health outcomes, given the symbiotics with the extensively hydrolyzed formula, there was less frequent wheezing and less noisy breathing. And very interestingly, the children in the symbiotic group also started to use asthma medication later than the control group. So definite effects on the microbiome, and then some of the clinical outcomes studied too. And you can also refer to these two figures on the slide to show the differences. Then what about just normal infant food? So we talked about delivery option, we talked about environmental exposure, we talked about drug exposure, we talked about supplementing infant formula with symbiotics. So would what an infant eat necessarily change their gut microbiome. And very interestingly, this is data from Lawson et al. Showing that as the infant is moving on to family food, we also see an increase in gut microbiome diversity measured by the Shannon index. And you can see the increase in gut microbiome diversity as the infants are eating more family food, eating more fiber, eating more protein. But interestingly, as the fat intake increased, the gut microbiome diversity came down, and the effect of carbohydrate eating or health conscious food didn't have such noticeable effect as just having family food and a higher fiber intake. So food diversity, increasing the variety of food an infant is eating in infancy, we've seen it can affect the gut microbiome. Is it relevant to allergy outcomes at all? So this is my own data from the Isle of Wight, where we clearly showed as the number of foods increased by 6 months of age, so did the odds of developing food allergy came down. I'll just go back to the figure at the slide before. And we also showed that as the number of allergenic foods increased over the first 12 months, so did the odds of developing food allergy over the first 10 years of life came down. But I think the next slide summarizes it a little bit better, where we can say that there was a consistent pattern of increased diet diversity, so more foods and in the first year of life, and reduced food allergy outcome. And for each additional food given by 6 months of age, the odds of developing food allergy over the first 10 years of life came down with about 11%. And for each additional food allergen scored by 12 months, so for each additional food allergen given by 12 months, the odds of developing food allergy came down with about 1/3. So definitely important to focus on method of delivery. Don't use drugs that we don't need to use. Think about environmental exposure. Think about breastfeeding, which formula to choose if mother chooses not to breastfeed or need to have a supplement feed. But then we need to go further to think about infant diet, getting babies to love food, increase their variety of food over the first year of life. Don't be scared to feed food allergens, and get the children on a family diet, hopefully a more healthy family diet by 12 months of age. And this is data from Caroline Ruday's group, which I just want to highlight before I summarize, where they showed that just increasing the diversity of cheese intake in the first year of life can lead to reduced atopic dermatitis and food allergy. And so I always jokingly say that, of course, we can do this kind of study in Switzerland, perhaps somewhere else in Europe, because the children do eat different kinds of cheese, but perhaps in a country where the cheese consumption is focused on one type of cheese, such as in America, this may be a more difficult study to do. In the same group of children, they also showed that early life diet, particularly a diet characterized with more fish, more yogurt, and more fruit and vegetables, was associated with reduced atopic dermatitis, asthma, and food allergy. And when they looked at the microbiome of these infants, they found that children who consumed the diet, which I've just described, had more butyrate levels and therefore more T regulatory cells and their associated cytokines. So definitely, if everything else goes wrong, we can perhaps still try to fix or prevent the development of particularly food allergy in early life by focusing on weaning and the weaning diet. So I would just like to summarize to say, I hope I've showed you that gut microbiome diversity is important. We want to have lots of different bacteria that's beneficial for the prevention of many disease outcomes and promote positive health outcomes in infants. We may not be exactly clear what the ideal diversity looks like, but we have an idea that perhaps bifidobacteria is associated with less disease outcomes, particularly allergic disease outcomes, and that Clostridium difficile may actually increase the risk. We need to focus on pre, pro, and symbiotics because they feed the gut microbiome and may have associated clinical benefits, particularly in disease prevention and particularly in those with increased risk of allergic outcomes. The gut microbiome interacts with the immune system and may prevent disease, which is really why we're trying to focus on the gut microbiome. And early life feeding, as well as environmental factors, play an important role to create and establish a diverse gut microbiome. So I would like to thank you, and thanks all my collaborators across the world, my IACI collaborators, my Isle of Wight collaborators, and my local collaborators here in Denver, Colorado. I look forward to further discussions during the question and answer session.
Hello, it is my great pleasure to discuss the clinical benefits of symbiotics beyond the dietary management of infants with cow's milk allergy. My disclosures that are relevant to this talk is that I'm receiving honorarium for this presentation from Nutricia, and I was a recipient of the research grant and was a site PI, principal investigator for the clinical study, PRESTO trial of amino acid-based formula with symbiotics that I will present the results during this webinar. So the objectives are to describe the role of gut microbiota in food allergy and to describe the effects of a hypoallergenic amino acid-based formula with symbiotics on development of tolerance to cow's milk and infections in infants and young children with cow's milk protein allergy. We all know that early life is a critical time for the development of the gut microbiota and maturation of the immune system, and that the disruption of the microbiota and immune system development in early life has potential later life health consequences on immune disorders such as non-communicable diseases that may emerge in teenage years and as well as in adulthood. So during pregnancy and infancy and childhood, there is a development of microbiota and immune system, and this will be, any disruption will be affecting the frequency of infections and atopic allergic diseases in childhood, such as food allergy and asthma. There is a narrow window of opportunity for microbiota modulation in infancy. So to really establish a footprint of a healthy microbiota, we have to start early. There is some development of microbiota during pregnancy, although there's a minimal exposure via placenta, but there has been documented effect of maternal factors such as stress, diet in late pregnancy, obesity status, so body weight, smoking, and socioeconomic status that may have an effect on infant microbiota. Obviously, mode of delivery, such as vaginal versus cesarean section, and gestational age are also big factors for that modulate microbiota. In early infancy, the critical factors are the diet. So is this infant breastfed versus formula fed, as well as use of antibiotics, antipyretics, and importantly, proton pump inhibitors. In late infancy, the infant gut microbiota is affected by the introduction of solid foods, which introduce various insoluble, digestible carbohydrates, as well as, again, use of antibiotics and proton pump inhibitors. So these are very important modulators, but there are also others that have been identified through epidemiologic studies, such as cultural factors, geographical location, family size, and socioeconomic status, as well as standard of sanitation, that will have the impact on development of gut microbiota in an infant. I want to spend some time discussing human milk and its role in protection from infectious diseases. We know that breastfeeding is recommended by World Health Organization as the optimal nutrition for infants. And breast milk, in addition to nutrients, contains many immunomodulatory components that may contribute to reduced risk of infectious diseases and reduced frequency of gastrointestinal infections, otitis media, as well as respiratory tract infections. And infection-induced wheezing was observed in exclusively breastfed infants even after several years after termination of breastfeeding. And this can be attributed to the presence of secretory IgA in breast milk for anti-inflammatory response, for regulation of gut permeability. So the other important factors in breast milk are antimicrobial factors for gut maturation and innate and acquired immune development, such as cytokines, TGF-beta, interleukin-10, as well as growth factors, as well as prebiotics. So lactose is a prebiotic that is present in human breast milk as a second most common or most abundant solid ingredient, as well as human milk oligosaccharides that are preventing microbial attachment to the mucosa and protecting against infections, in addition to lactoferrin blocking the inflammatory cytokines. And it has been recognized for some time now that infants with cow's milk allergy often have an altered gut microbiota compared to healthy breastfed infants. Importantly, this can be observed before development of cow's milk allergy. So dysbiosis may precede development of cow's milk allergy. So in healthy infants that are fed with who are breastfed, the healthy bacteria that are present and predominate in the gut are Bifidobacterium species, Escherichia coli, Fecalibacterium prausnitzii, and Lactobacillus species. However, when there is a dysbiosis, there is a more adult-like pathogenic bacteria present in the gut, and these bacteria include Enterococcus faecalis, Methanobrevibacter, Clostridium difficile, and Campylobacter. And it has been noticed that this predisposition to dysbiosis that may be present in infants who are exclusively breastfed may be attributed to the differences in the composition of human milk oligosaccharides of the mothers who have infants who will go on to develop cow's milk allergy compared to those non-allergic infants. Breast milk is a gold standard for nutrition and is also an ultimate symbiotic. So breast milk is not sterile. It does contain live bacteria as well as prebiotic oligosaccharides as well as lactose that stimulate a gut microbiota and immune system. And in the past, the hypoallergenic formulas that were used for the management of cow's milk allergy were lacking this microbiota stimulating factor. So as much as they were made to resemble the human milk, breast milk, our knowledge was lacking of the importance of those microbiota stimulating factors. So human milk contains live bacteria, human milk oligosaccharides, as well as lactose, a main carbohydrate, and traditional extensively hydrolyzed formula had no bacteria, no oligosaccharides, and only small amount of lactose. And in the amino acid-based formulas, there was no lactose in addition to lack of bacteria and oligosaccharides. So this was really a major difference because the human milk is rich in factors that stimulate gut microbiota and immune system development, whereas the formulas that we're using for treatment of cow's milk allergy had really very limited or complete lack of stimulation of the gut microbiota. So really this understanding of the importance of gut microbiota has led to the development of formulas that emulate human milk. So the formula that was used in our studies was a amino acid based formula that contained a symbiotic blend of pre and probiotics, and it was designed to mimic the immunomodulatory components of human milk to target gut microbiota and to correct dysbiosis. So the prebiotic in this formula were either short-chain galacto-oligosaccharides and long-chain fructo-oligosaccharides in 9:1 ratio, or short-chain fructo-oligosaccharides and long-chain fructo-oligosaccharides 9:1 ratio. So this prebiotic mimics the levels and functionality of human milk oligosaccharides. It has been validated by over 30 clinical studies in infants in terms of the safety. And prebiotics have been acknowledged by the World Allergy Organization guidelines as for their role in the prevention of allergic diseases. The probiotic that is included is Bifidobacterium breve M16V. This is the most common bifidobacteria species in human milk, as well as in the gut of healthy breastfed infants. So this is why it was selected as an ingredient, as well for its ability to reduce allergic responses that has been shown in preclinical and clinical studies. Furthermore, it has a long history of safe use even in premature infants and is free from the main food allergens such as cow's milk proteins. This combination of prebiotic and probiotic works synergistically to target the gut microbiota dysbiosis. 3 randomized clinical trials have demonstrated that the synbiotics are capable of rebalancing the gut microbiota in infants with cow's milk allergy and bringing it closer to that of healthy breastfed infants. Gut microbiota of children with cow's milk allergy is characterized by dysbiosis, so this disrupted composition. But when those children are fed with this formula of synbiotics, there was a beneficial change observed in terms of increase in the amount of bifidobacteria and decrease in Escherichia coli and Clostridial species. And this was sort of shifted towards the phenotype of gut microbiota of healthy breastfed infants. In addition, there were 3 trials. So first trial was done in infants with cow's milk allergy, mostly IG mediated. Those infants were fed with amino acid formula with synbiotics, and there was an increased levels of bifidobacteria and reduced pathogenic bacteria levels. In the trial of non-IG mediated cow's milk allergy, when the same formula was used, the levels of fecal bacteria resembled the healthy breastfed infants with increased levels of bifidobacteria in the active group infants, and the effects were seen up to 26 weeks. So this is quite remarkable. And in the third trial, the Presto trial, we have used this amino acid formula with synbiotics, and we observed increasing bifidobacteria as well as similarly reduced Escherichia species and Clostridial bacteria levels. So let's move on to the Presto study, which was a multi-center randomized controlled clinical trial that was aimed to evaluate tolerance development in infants with IG-mediated cow's milk allergy receiving amino acid-based formula, including specific synbiotics. The objectives of the study, the primary objectives were to evaluate development of tolerance to cow's milk at 12 months of dietary management with an amino acid-based formula with synbiotics and without synbiotics, as measured by Milk Challenge, double-blind, placebo-controlled food challenge in infants with IG-mediated cow's milk allergy that were being fed with the control or test product. The secondary objectives were to measure development of tolerance to cow's milk in early childhood, to evaluate maturation of the gastrointestinal immune system in IG-mediated cow's milk allergy, to confirm that the test product is effective in the dietary management of cow's milk allergy, to evaluate the occurrence of non-food allergies, and to evaluate safety through adverse event monitoring. We have enrolled 169 infants between 0 to 13 months with IG-mediated cow's milk allergy that was confirmed by an oral food challenge or through a recent convincing history of anaphylactic reaction. The study took place in 18 study sites in 6 countries in Europe, US, and in Asia. Those infants were randomized 1-to-1 to either receive symbiotic containing amino acid formula or to a control formula that was lacking the symbiotic. So infants were fed with this formula for 6 months, and after 6 months, they were then continued to be followed up but no longer fed with the study product. And at this point, we have a 24-month follow-up, and we continue following those infants for longer periods of time. So the primary parameter was evaluated after 6 months of feeding with the formula, with the study formula, and it was a tolerance to cow's milk assessed by the double-blind placebo-controlled food challenge after 12 months of dietary intervention. And the secondary parameters were tolerance to cow's milk after 24 months and 36 months, presence of allergic symptoms, skin prick test results, immune markers, as well as development of other non-food allergies. And these parameters are still being evaluated. So after 12 months of intervention, approximately 50% of children receiving amino acid formula developed cow's milk tolerance. However, you can see there was no difference, no statistically significant difference between the tests, so symbiotic and control, no symbiotic amino acid formula fed infants at 12 months and at 24 months. However, I want to point out that this is a high level of tolerance development if the infants fed with amino acid formula, and this is comparable to the data from the literature reporting the rates of tolerance development in infants fed with extensively hydrolyzed formulas. However, we've observed a very interesting fact that in terms of safety, fewer infants in the test group required hospitalization due to infection. So there was no difference in terms of overall adverse event frequency or gastrointestinal disorders or infections and infestations between the two groups or any serious adverse events. However, there was a statistically significant difference in terms of the infections leading to hospitalizations, with 9% observed in the test formula group versus 20% in the control formula group, suggesting that addition of symbiotic has a protective effect against the infections. And really excitingly, this follows a trend that was observed in prior studies. So in those 3 randomized clinical trials with amino acid formula with symbiotics, there was a consistent reduction in infections and antibiotic usage in infants fed with the symbiotic formula. So in the first study that was conducted in the US with the infants with the Ig-mediated cow's milk allergy, you can see significant difference in the infections. And I just want to point out that the blue bar shows the control formula without symbiotics, whereas the pink bar represents the amino acid formula with symbiotics. So you can see significant reduction in infections. There was also reduction in the use of antibiotics, and particularly in the use of amoxicillin in that trial. In the study with the non-Ig mediated cow's milk allergy, there were fewer ear infections, as well as the lower usage of anti-infective agents in the infants fed with the formula with symbiotic. And finally, in the PRESTO trial, where we have enrolled patients with severe Ig mediated cow's milk allergies, we've observed fewer infections requiring hospitalizations in the infants fed with the amino acid formula with symbiotics, which I think, it's a very clinically relevant observation. So the conclusions from our studies was that about half of the children developed oral tolerance to cow's milk within 12 months of intervention. And this is in line with the previously reported rates of tolerance development of infants fed with the extensively hydrolyzed formulas. Additional 13% developed oral tolerance within 24 months. However, there's no difference in oral oral tolerance development between children who received an amino acid-based formula with or without symbiotics. However, children receiving an amino acid-based formula with symbiotics required fewer hospitalization due to infections, and this is in line, consistent with the two other randomized clinical trial of the same formula of symbiotics in infants with IgE as well as a non-IgE mediated cow's milk allergy. So what is the clinical significance? And that's particularly relevant to me as a food allergist because there is a strong relationship between atopy and increased susceptibility to infection. So infants with who are destined to develop, atopic infants who are destined to develop food allergy, atopic dermatitis, and other conditions are born with the strong Th2 skewing at birth. And we know that all infants are born with Th2 predominant immune system to prevent rejection of infancy and reactions graft versus host from the fetus against the mother. So they are, the Th1 responses are really very immature, and this translates into increased susceptibility to infections, as well as development of atopy. And this is further modulated by presence of gut dysbiosis that may be affected by mode of delivery or use of antibiotics because of the complications, as well as a type of infant feeding with formula feeding, lacking the immunomodulatory components that are present in breast milk. And all of these factors sort of converge to increase the risk of atopy and increased infections in the first 3 years of life in atopic infants. And we know that infants with atopy have a high frequency of infections, so average 14 infections during the first 3 years of life, which translates into a higher use of antibiotics for ear infections as well as respiratory tract infections among those infants, with the most frequently used drugs being amoxicillin and penicillin. So I'll just want to share with you a typical infant patient in the pediatric allergy clinic. This is Bobby, who was born at full term without complications. He was formula fed since birth because his mom couldn't breastfeed. And at 6 weeks, he was noted to have an itchy and oozy rash and was diagnosed with atopic dermatitis. In addition to atopic dermatitis, he wasn't gaining weight. His rash was becoming more severe. So at 4 months of age, he was diagnosed with cow's milk allergy, and he was switched to a hypoallergenic, extensively hydrolyzed formula, with ultimately transitioning to amino acid formula due to ongoing symptoms and poor weight gain. And he finally, resolved his symptoms on amino acid formula. However, between ages 6 to 12 months, Bobby had 3 episodes of otitis media that were treated with antibiotics, specifically amoxicillin, and he underwent placement of ear tubes at 13 months of age. He also developed the first episode of wheezing at the age of 15 months. And his parents, having read on the importance of microbiota, are now asking about the probiotic supplementation. And you can easily, understand why, having this probiotic supplementation early on in life might have prevented the use of antibiotics and infections in a patient like Bobby, and would be very clinically relevant. So in summary, the new generation of hypoallergenic infant formulas with symbiotics replicates the immunomodulatory effects of breast milk. And this is very, very important because to date, those excellent products or excellent hypoallergenic formulas were lacking those very important components that are present in breast milk. We have evidence that amino acid formula with symbiotics promotes colonization of the infant gut with beneficial bacterial communities, and that amino acid formula with symbiotics has a consistent, as seen in 3 clinical trials, protective effect from infections in early life. So fewer infections, fewer use of antibiotics, and this has been seen consistently consistently. And this effect is of special relevance for atopic infants who are born with predisposition to develop frequent infections due to the strong immaturity of the Th1 responses and strong Th2 skewing. And in closing, I want to thank all of the investigators who participate in the PRESTO trial, as well as the Dano Nutrition research team for their support for that great study. Thank you very much.
We have a few questions, and obviously you've raised some very important points. The questions deal with the idea of, is this standardized all over the world? For example, probiotics and prebiotics, do all the world allergy organizations endorse it? I know that there was mentioned that this is guidelines for WAO, but one person asked if EAACI had these guidelines and other organizations, and do we need a multi-country standardized trial and standardization of care across the world? So I guess the PRESTO study raises some of that. So I'd like each of you to tackle the issue of, do we have standards? Is this standard of care, and how can it get standardized? Anya first.
Yes, so I think it's a great question, and obviously, those presentations are raising those questions. But we have a lot of information, we have a lot of evidence, but the evidence that we have, and it's a very strong rationale for using pre and probiotics, right? But to get to the point of issuing guidelines, I think we're still short on having the definitive clinical trials. So I think we would need to have more, high quality evidence to sort of issue like a sweeping, worldwide approach in terms of supplementation. I mean, obviously, we all agree that diverse diet and breastfeeding, so sort of natural sources of pre and probiotics and immunomodulatory factors in breast milk are the right way to go. But in terms of saying, oh, this is how much of this pre or probiotic and this particular strain you have to take to achieve this desired effect, I don't think we are there. But one thing I would like to add to this is that in terms, well, it's a huge, there's a huge diversity in how pre and probiotics are manufactured. They are there as supplements, and you have to be careful what are the sources of those pre and probiotics, and whether, particularly when you're using them for infants with food allergy or cow's milk protein allergy, because there could be some cross-contamination. So, pre and probiotics used within the formula, the hypoallergen formula, are tested and they are proven and guaranteed that they are not, containing other allergens. So I think this is a very important point to give, and they're safe. Even if the formula was the only source of nutrition for an infant, the amount of the pre and probiotic within this formula is absolutely appropriate and is safe and supports normal growth in those infants. But when it comes to supplements, I don't think we are there yet.
Okay, thank you, Anya. Karina, do you have comments on ideas of standardization, guidelines?
Yes, I think I'll take it from two aspects. So the one is the pre and probiotic aspect. The latest IACI prevention guidelines are open for consultation. If you follow me on Twitter, I've posted the link. You can have a look to see what we've said in the preliminary guideline, or the one that's open for consultation about pre and probiotics. I know that IACI is also working on a guideline focusing just on infant formula. I'm not part of that group. It's not published, but do keep an eye out. I'm not aware of any official guidelines from the American College or the American Academy. So I think answering that. Then in terms of standardizing infant formula, I think, this goes beyond just pre and probiotic content. The nutritional content of infant formula, the FDA has got very different guidelines in terms of nutritional composition of infant formula compared to the European guidelines. And I can tell you this because as a dietitian, what we boringly do is we forever creating these tables comparing nutrient content of different formulas across the world. And it is very, very different. So I don't know if we're ever going to get to the point where we have just one combined standard, but the standards differ on many different levels.
That's very, very insightful and very helpful. And we'll go to your Twitter or whatever you've posted. I wanted to ask myself, but first, just a very short question. One of you could take this. Are these prebiotics, the FOS and GOS, available for supplementation independent of formula? Does anybody know that?
I don't think they are. No.
Okay. Okay.
Not to the best of my knowledge. They're only available as an ingredient.
And I'd like to ask
fruit
you,
and vegetables.
Yeah.
Yeah, family food. Anya, I'd like to ask you, beyond the fact that of Th2 and Th1 and that intimation of the immune mechanism, I can ask Karina too, what is the immune mechanism of action or mechanisms of action? At what levels do they work, if they work, to change IgE-mediated disease or even non-IgE-mediated? And I'll add to that that the data didn't really show difference from natural course in terms of tolerance or in terms of change, but they did show a change in immunity. So what is the mechanism of action?
Well, I think our results from the Presti study, right, would suggest that the role of pre and probiotics would be much more important for prevention than for treatment, of established allergy. Our study focused on the Ig-mediated cow's milk allergy, and we know that this is a more difficult allergy to outgrow. So, but there is a good evidence that there is an interaction with the immune system. So the short chain, the oligosaccharides that are included as prebiotics are being digested by the beneficial bacteria that already reside, and the bacteria that you are providing as probiotics within the formula. They're being digested to short chain fatty acids, and the short chain.
But I'm trying to get at what happens to the immune system. Like,
that's what
Karina talked about the T cells.
Right, no, but that's what happens is that, butyrate, for instance, has been shown to interact with the receptors on the surface of the Tregs, of the T cells in the gut, and skew them towards production of IL-10, so generation of tolerance. So, in that regard, this is very important for both Ig and non-Ig mediated food allergy, because we know that this would, sort of set up the stage for development of normal oral tolerance in the gut.
Okay, thank you. Karina, related to that, and in addition to that, would butyrate be a factor that would propitiate the development of Tregs?
Well, I think so, based on the research we have at the moment. I've just put a grant in, which, fingers crossed I'll get funded, to look at this particular effect and also particular fibers affecting butyrate content. Because again, being the dietitian, the only food that really contains butyrate is ghee, so we don't really want to turn the whole world into a ghee consumption world, because there's a lot of saturated fats, and when you fry foods, you have H's. Well, you're probably good on your T-Rex this morning, but, and also frying foods in ghee produce advanced glycation end products, which has got another issue. So I think, and I hope the answer lies in butyrate, but exactly how we best manipulate the gut to have optimum levels of butyrate, I think we just don't know yet.
Okay, excellent. We have one more minute, barely. For Dr. Anya, do you recommend starting probiotics routinely in eczema patients? And what is the type and dose you recommend? Obviously, someone wants a guideline.
Well, that's the thing, like there's no guideline, and I don't recommend and I don't start it routinely. However, I'll share with you that I would definitely consider, using the probiotic, as a trial of in an infant that has, eczema, has been born via C-section. Like you've shown, your data has shown that, those infants have, high risk of having a, dysbiosis. But the truth be told, I don't know exactly what's the dose or what is the right probiotic for this infant. So it's a trial, and and then the reason also I don't use it routinely, not that I'm worried about the side effects, but it's a cost. It's a cost issue.
Sure.
So those preparations tend to be expensive. So it's okay to try, but with the understanding of the limitations that we don't know exactly what's the dose and what's the right probiotic for that particular infant.
Excellent.
I can quickly add, in the US, the only probiotic that I'm aware of that contains lactobacillus as well as bifidobacteria that's guaranteed to be cow's milk protein free is about $40 a month. It's a huge cost implication.
It's expensive, so it's not available to everybody, and it's not covered by the
Anyway, I was enlightened by your talks. I really enjoyed the interaction, even though it's Zoom and not in person. We are 2 minutes over the hour, but we started a bit late. So I wanted to thank you, Karina, and you, Anya, and the organizers and our sponsor for this wonderful session. And I think we have a lot of work to do, but a lot of very interesting work to work upon. Thank you very much.